This video, I'm going to go through section 4.6, which is titled Additional Principles of Experimental Design. So they talk about how a well-designed experiment has 4 components. So of those 4 components, it's comparison, control, random assignment, and replication. And so they spent most of their time talking about comparison. So you should expect to see a lot more quiz questions on that section too. So comparison is when you're placing the experimental units or subjects into groups so that you can compare those groups. A control group is a group that provides the baseline for comparing effects of treatment. So this means that the control group can either have an inactive treatment, so placebo, an active treatment, maybe at a different dosage or a different level, or they could receive no treatment at all. Having a control group is not required if all treatments are applied to every experimental unit. So for example, if we're talking about maybe like changing the amount of times you water a plant, right? If I'm doing the same thing for every plant, then I don't have to have a control group. So in this example, we have a doctor that wants to determine if a new medication for cholesterol is more effective than the previous medication. The doctor informed his patients of the new medication and asked them if they would like to participate in a drug trial to test the effectiveness of the new cholesterol medicine compared to the current medications. So 25 patients who were currently taking the medication for cholesterol volunteered and were given the new medication. After 2 months, the patient's cholesterol levels are checked. So notice that there's only one group here, the group that receives the new medication. He doesn't have another group that's taking the original medication where he can compare the results. So because the doctor did not have a different random sample of patients take the original medication, the effectiveness of the new medicine will not be able to be determined because we don't have anything to compare those cholesterol levels to. So that's why comparison is important, so that you can see, okay, what happened with the old medication? What's our data from that? So that I can compare the data to the new medication and see if those levels have improved. One thing that's important to know about experiments is the placebo effect. So placebo effect is when subjects can respond favorably to any treatment, even in an active one. So for example, to test if vitamin C actually makes people feel better, a vitamin company decides to run a 5-day study where they give one group of 100 sick patients vitamin C pills and another group of 100 sick people placebo pills, and monitored another group of 100 sick people who took no pills. So if we think about it, we have 3 different groups. We have vitamin C pills, we have placebo pills, and we have no pills. And so they have the same number of participants. At the end of the 5-day experiment, 90 participants in the vitamin C group reported feeling better, 30 in the no pill group felt better, and 50 participants in the placebo group felt better in a 5-day period. So in theory, these numbers should be the same because they had the same medicine. So the fact that more people felt better in the placebo group is an example of the placebo effect. Okay, so they're thinking that this thing's going to make them feel better, helps them essentially trick their bodies into thinking that they felt better when they might not have actually. The way that we can reduce the placebo effect is by blind experiments. So a single blind experiment is where either the subjects or those measuring the response variable don't know which subjects received the treatment. So that would be an example of this, where they don't know that they got a placebo, right? So that's an example of a single blind experiment. A double blind would make it our data even better, where neither group knows which treatment the subject is receiving. So that means that the person administering the pills or collecting their data for whatever they're studying doesn't know whether that subject got the placebo, got the vitamin C. And so that is a way to reduce the placebo effect. So in this example, we've got a new cream was developed to reduce irritation caused by poison ivy. To test the effectiveness, the researchers put an ad online asking for volunteers to participate in a study. 100 subjects applied. They were informed that one group would receive the new cream and the other group would receive a cream with no active ingredient. All 100 subjects were exposed to poison ivy. 50 were assigned the group with the new cream. 50 were assigned the group with the cream with no active ingredient, so that's the placebo. After 3 days, the subject's level of irritation was measured. The technicians who surveyed the subject's level of irritation, as well as the subjects who did not know which group received the treatment. So this is an example of a double-blind study, because both of them didn't know. If only one of those two groups didn't know, then that would be a single blind study. So the next characteristic is control. So it's not a control group, though we're talking about the same word. Control means, in this case, keeping other variables constant for all experimental units. For example, you would want to make sure that the people that you are studying are within the same age range, maybe within the same weight range, that they're all non-smokers. You would ask them not to make any other lifestyle changes during the time that they're doing the study, because those things are factors that could factor into whatever you're studying. So in an example of maintaining control, a biology student wanted to determine if using fertilizer would promote plant growth. 100 plants were randomly assigned to one of two groups, one that used the fertilizer and one that did not use the fertilizer. So the goal is that that's the only thing that's different, so that I can conclude that any of my results are only related to the fertilizer. I was gonna say refrigerator. Each plant was planted in the same amount of soil, given the same amount of water, and the same amount of light. So they're controlling the situation so that those things don't affect the growth of the plant. Sure, it might be able to. Every plant could grow differently, but they're trying to control the situation enough so that it would not affect the data, so that it's only the fertilizer that's the difference between these plants. The third component of a well-designed experiment is random assignment. And so what's important is that an experimental process experimental units are assigned to a treatment using a chance process. So the experimental units, or subjects if they're people, are randomly assigned to which treatment group, right? So this allows the variability of the experimental unit's characteristics that may affect the response variable to be evenly distributed among groups to ensure the results are from the treatment and not those characteristics. So if there are things that they can't control, random assignment helps spread those uncontrollable characteristics across the treatment groups. Okay, so if it's asking how this is an example of something being random, you're looking at the idea that they're evenly distributed among groups to ensure the results are from the treatment and not those characteristics. Okay, so that's the big idea with random assignment, is that they're evenly distributed among the groups, so that it helps with something that they can't control. The final component is replication. So replication is giving each treatment enough experimental units so an observed difference in the effects of the treatment can be distinguished from the chance variation due to random assignment. Okay, so again, this is related back down to the random assignment. So the more samples that I have, the larger sample that I have, the better I can compare the differences. Okay, so if I have a small sample, those characteristics that influence the response variable could have a higher influence, whereas if the larger my sample is, that average is going to get closer and closer and closer to the real average. So in this last example, a student wants to determine if different types of soil, A and B, affect the propagation of African violets. 100 leaves from the same variety of African violet were selected. The leaves were then randomly assigned to 50 pots with type A soil, and the remaining leaves were put into 50 pots with type B soil. All the pots were given the same amount of water and arranged in 10 rows of 10 pots near the window for light. The 50 pots with type A soil were placed closest to the window. At the end of 2 months, the pots with type A soil had the greatest number of pots with new growth. So the student concluded that soil type A was more effective than soil type B. Is this a valid conclusion? So our options are yes, because the leaves were randomly assigned. This evenly distributed the variability in the leaves. So that's true. That is a good part of this experiment, that the leaves were randomly assigned. Then our second option is yes, because the amount of water and light were controlled. This may have reduced the variability in response. Therefore, type A soil is more effective. And so if we think about that, the water and light weren't really controlled because all of the pots with type A soil were placed closest to the window. So that's definitely not true, right? The light was not controlled. And so no, because the pots with type A soil were closest to the window, they received more light, which may have led to the new growth. And then finally, the other option is no, because African violets were used, replication was not properly implemented. So this isn't true. You want to use the same type of plant in order to compare, right? Because different plants grow at different rates or propagate at different rates. So the correct answer here is that no, because those pots with type A soil were closest to the window, they actually got more light than those with type B. Okay, so those 4 components, replication, random assignment, control, and comparison, are what create a well-designed experiment.